Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on enzymes. So far in this unit, you have learned that enzymes are proteins that catalyze chemical reactions. You have studied the properties of enzymes, and you have learned how to name and classify them. You even had a chance to act as real biologists and present your findings on enzyme naming to the class. Today's lesson will be a review of what you have learned so far. If you are ready, students, let us begin. You have already learned that an enzyme is a globular protein with a uniquely shaped active site. It acts as a biological catalyst for a specific reaction, but it remains unaltered by the reaction. This definition provides us with 3 of the 5 properties of enzymes. As a review, please try to list all 5 properties. I will see you in a few minutes to go over the answers.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Hello again, students. Were you able to recall all 5 properties of enzymes? From the definition, we know that all enzymes are proteins. We also know that they are biological catalysts. They are able to speed up chemical reactions. Enzymes are also specific. Because of their uniquely shaped active sites, enzymes can only bind with a specific substrate or substrate combination. This means that only one possible reaction can be catalyzed. It only takes a small amount of enzyme to bring about a change in a large amount of its substrate. Finally, enzymes are affected by both pH and temperature. Higher temperatures can destroy enzymes by changing the shape of their active sites. pH that is too high or too low can also affect the shape of an enzyme's active site, thus preventing it from binding with its substrate. If you were able to list all 5 properties, you have remembered well. Good work, students. You have also studied the classification and naming of enzymes. I find the history of naming enzymes to be quite interesting. By the 1950s, the number of known enzymes was increasing rapidly. The naming of enzymes by individuals proved to be quite confusing. Many of the names conveyed little or no idea of the nature of the reactions they catalyzed. Some enzymes were named based on the source from which they were first identified, such as papain from papaya. Other enzymes were named by adding ASE to part of the name of the substrate, such as lipase and sucrase. Still, others ended with IN to indicate that they were basically proteins. Some examples of these are pepsin and trypsin. In order to regulate the naming of enzymes, the International Commission on Enzymes decided to produce a systematic way of naming enzymes based on the ways in which the enzymes act. The Commission began by organizing the enzymes based on the types of reactions they catalyze. There are 6 classes of enzymes in this system of nomenclature. Do you recall them, students? Please work together with a classmate. Write down the names of the 6 classes of enzymes and a brief description of the types of reactions they catalyze. You may begin.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. Did you work well with your partner? Let us look at the answers together. The first group is called Oxido Reductase. These enzymes catalyze reactions in which hydrogen or oxygen atoms or electrons are transferred from one substrate to another. The second group is called transferase. These are enzymes that catalyze reactions in which a specific group, such as a methyl, is transferred from one substrate to another. The third group of enzymes is called hydrolase. These enzymes catalyze the hydrolysis of a substrate. The fourth group is called isomerase. Enzymes in this group catalyze reactions in which the molecular form of a substrate is changed. The fifth group is called lyase. These are enzymes that catalyze the non-hydrolytic removal or addition of a group to a substrate. The sixth group of enzymes is called ligase. These enzymes catalyze reactions in which two molecules are joined together by the formation of new bonds. If you were able to list all 6 groups, you and your partner did very well. In the systematic naming of enzymes, an enzyme is first classified into one of these 6 groups. From these groups, they are classified further into subclasses and subclasses. Within the subclasses, each enzyme has a number. Let us look at an example together. Here is an enzyme named EC31173. The EC stands for Enzyme Commission. 3 tells us that this enzyme is a hydrolase. 1 tells us that the enzyme is a subclass that acts on ester bonds. The second one indicates that the subclass is the carboxylic ester hydrolase. Finally, 73 is the number assigned to this particular enzyme, which is ferrolyl esterase. There are far too many enzymes for us to be able to remember all of the subclasses or even the subclasses. This is why the Enzyme Commission maintains an accurate and systematic database of names. Any additions or changes to the database must be approved by the Commission. Students, today we have reviewed the properties of enzymes. We have also discussed the ways in which they are classified and named. In our next lesson, we will focus our attention on the function of enzymes. Until then, thank you, teacher. Thank you, students.